Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Novartis, Allergan, Janssen, Biogen, AGTC, MeiraGTx, and the SickKids Foundation. We'd also like to thank Accessible Media Inc., our national accessibility partner. Everyone should download the AMI app, which is a fabulous resource, and you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoyed this webinar and share your thoughts with us in the comment section below.
Thank you. So my work on hereditary eye diseases has been particularly in Newfoundland and Labrador, although many things, research studies that are done in one location can have implications for people elsewhere. Next slide. So this is the island of Newfoundland. Labrador is further north, but Newfoundland was settled primarily in the 17 and 1800s by immigrants from England and Ireland. When the original explorers came to North America, they reported rich cod stocks around Newfoundland, and that's really why people came. And so they settled in all the small coves and bays around the island. And really one ship would sail into one cove and the people would settle there. And over the generations, typically the descendants stayed in the same community. Family size was often quite large, 10, 12, 15 or even more children. And so there would be a cluster of people in one community that were closely related to each other, but quite different from a neighboring community. There were no roads, essentially. Everything was by sea, but people essentially were in their own outports. The next slide, please. And so because of, and you can click to get each line on. Because of this pattern of settlement in the large families, we may have clusters of rare genetic diseases that are rare in other circumstances like large cities. And as with any disease, they need a diagnosis and treatment, but they're also valuable for genetic research because of the number of people. And they tell us more about the clinical features of a condition. If you just have one person in a family, you just know what they're like. But if you have several brothers, sisters, cousins, you can see if it's all the same, what age does it start, how fast does it progress. And also, large clusters help you find the gene and mutation for a condition. So this research is, as well as being valuable just for genetics research, it's also improves diagnosis and helps with management. So if families are part of the research, they actually benefit in the end because of what is learned. Next slide, please. So we're talking about genetic diseases, and genes are our computer program. They determine how our body develops and how each part of our body works, so that there are genes that have to do with the brain, the heart, the eyes, the ears, the muscles, and so on. And what we know now is that there are many, many genes working together for each part. So there's hundreds of different genes that have to do with eye structure and function. Next, please. And genes are made of DNA. And genes are a long coil, a double coil. And DNA has 4 bases, A, T, G, and C, and they're like the alphabet of DNA. English language has 26 letters. DNA has 4 letters, but it's the combinations of those 4 letters that determine how something called a protein is made, and the protein is really what is the structure of the body and causes the functioning of the body. But the DNA is the critical stored information. Next, please. So genetic diseases are caused by mistakes in our genes. And the map on the side of the screen shows just different clusters of genetic diseases. They're heart and eye and hereditary cancer and so on, but just many different hereditary diseases that are being studied. The eye that you see has an abnormality of the front of the eye, and that's seen in one large family that I studied. And then the other big pedigree, which is what we call the drawing of family members, how they're related to each other. That's a very large family with hereditary cancer. I won't talk about those today, but that's the other area that I have worked on. The next, please. So I said that these letters, the G, C, A, G, A, T, T, G, T, maybe what that order of that section of DNA should be. But if in that second code word, if you have a G in the middle instead of that A, that means the protein is changed. It seems like a very small change, but it can make a dramatic difference. So if the sentence in English was, a cat is on my bed, well, that's my cat Fluffy, that's great. But if there's a mistake and it says a rat is on my bed, I'm not so pleased about it. So next. And so this is just any alteration or mutation of a gene may cause human disease. So that very small mistake can be a mutation enough to cause a disease. Sometimes the mutation is larger. It's like a missing word or a missing sentence. But this is basically changes in DNA cause genetic diseases. Next, please. So I mentioned I grew up in Vancouver and went to the University of British Columbia and did a zoology degree. And then in the mid-1960s, I did my master's degree in Drosophila genetics. In those days, people knew that there were clusters of diseases in families, but DNA was barely known. It certainly, people didn't know anything about how to sequence DNA or find genes, but you could study other organisms, fruit fly, mice, dogs, and so on, to understand how conditions ran in families. And I was very interested, very inspired by the genetics. At the time, my husband completed his PhD in marine biology in 1967 and got a position in Newfoundland. So we drove here and have been here ever since. And at that time, the medical school had just started in 1968. It was only teaching in temporary buildings. But in 1978, the Health Sciences Center was actually built, and that housed the St. John's General Hospital and the medical school. And so this was my chance to go and talk to faculty members and physicians in the Health Sciences Center. And I met Dr. Gordon Johnson, who was an ophthalmologist originally from England, but he'd been in St. Anthony, the northern tip of Newfoundland, and had flown in for clinics in the rural hospitals in Labrador. And he was giving a seminar about some of that work in Labrador. And I talked to him afterwards and said I was interested in something in genetics. And he was interested in dedicating more time to seeing hereditary eye diseases. And fortunately, when we talked to the CNIB, they were interested in funding a clinic that was once a week, and initially at the hospital in St. John's, but then we began having clinics in rural hospitals. And this was before computers. So the CNIB had paper charts and they had filing cards, and it had the conditions, but it really talked just about, they were problems of the retina, and it might be a diabetic retinopathy or age-related, or it might be a genetic disease of the retina. They had no way of knowing how many people had genetic conditions. And so I reviewed all the records in order to sort out that question. Okay, next slide, please. So I mentioned on the right-hand side of the map, the red star is where St. John's is, and that's where we initially were having our clinics. But very frequently, I would say, is there anyone else with something similar to you? And they say, well, yes, there's my brother and sister, but they live in Twillingate. Or I've got several cousins and my aunt and uncle, but they live on the west coast of Newfoundland. And in those days, it was very difficult for people. The roads were poor. It was very difficult for people to travel into St. John's, a number of hours travel. And so we had the clinics in the rural hospitals to see other family members to determine if they really did have the same condition, and if so, what was the pattern of inheritance? Was it recessive, all in one generation, like brother and sister, or was it every generation, so a parent and grandparent and so on was affected, or was it all in males, and that would be an excellent type of inheritance? And you needed to know that if you're going to give any kind of advice to family members about risk to others in the family. So over the next few years, we had clinics in all of these locations mentioned or shown. And so the next slide, please. And when I finished the review of the CNIB records in 1985, almost 1/3 of those registered had a hereditary eye disorder, and nobody had predicted that. What you see is the front of the eye. When we talk about the eye, we say somebody has brown eyes or blue eyes. You're just talking about the front. But the real works of the eye are the inner surface, called the retina. And so the eye is like a globe or a ball, and it's that inner surface that's critical. And then there's a connection from the back of the eye to the optic nerve to the brain, because the brain interprets what you see. And any part of the eye could be involved in a hereditary disease, but the most significant ones are those of the retina. And one thing, as we were doing our clinics, we noticed that many of the children and teenagers were reported as doing poorly in school, but the poor vision had not been recognized. And at that time, the teacher wrote everything on the classroom. There was maybe quite poor mimeograph. If you couldn't read this information, you couldn't do well in any kind of testing. And so it was very important to recognize the poor vision and work from there. So next slide, please. So these are just within the hereditary conditions. There were the retinal dystrophies, RP, and others. Bardet-Biedl syndrome has RP, and the rod cone dystrophy, a certain type of RP. Stargardt disease is central macular, but juvenile, and then the types of albinism, which are pigment problems. And again, the map showing where some of these are. Next slide, please. So we realized it was very important to recognize and make an accurate diagnosis of hereditary eye disease. Because of the education, we saw that many people had done poorly because they couldn't see. Fortunately, the government in the 1980s started having itinerant teachers for the visually impaired, and they went to the individual schools and helped with visual aids, which initially was large print and magnifying glasses, but soon when computers became available, these young people were the first to get them. And now, of course, computer technology can really do wonders. But if you didn't have good education, then it was very difficult to have good employment, and you really needed career planning. If somebody thought, had hoped to be a pilot, well, that wasn't possible, but there's other very good types of employment if you were helped to find them. Family and social life were affected. The kids couldn't go out and play in Newfoundland on the frozen ponds. They played hockey. Well, if you can't see the puck, you can't play. You couldn't drive. It was hard to get together with friends. And then the other thing was risk of recurrence, and you needed to know how it fit in the family, and you needed to know whether it was a stationary condition, whether it was poor vision but stay the same or get worse. So all of this was important. And now the added thing is, as was mentioned in the introduction, treatments are being developed for some hereditary eye diseases, and you need to know which exact condition you have. So next, please. So this is a young boy with the condition called ocular albinism. So he has, albinism is lack of pigment, and there needs to be pigment in the retina. He has a very pale retina and had very poor vision right from birth. And he was part of that very large pedigree on the top right. Dr. Janssen had seen some of these people before, and we went through communities all in the Northeast coast to see really all of the people in this family. It's X-linked, and so it was primarily the men and boys were affected from a young age. Girls could be carriers, and so they had the possibility of having affected sons. So we wanted to provide the information to everyone. And because this was a very large family, when it came possible to be searching for the gene, this family was part of the studies to find the ocular albinism gene. And later there was another family with the same condition on the south coast of Newfoundland, had a different mutation in the same gene, but it is worked out as well. Next, please. There's also the condition oculocutaneous albinism, where besides the back of the eye lacking pigment, the hair and skin. And there were 4 families on the Northern Peninsula and Southern Labrador, and actually 3 different genetic defects. So if you're thinking, one family may in the future have a treatment, you can't automatically say, well, the family in the next community does, because what we found here was it was a different gene. Next slide, please. Another person we saw very early on, an infant was born in 1979, and he had completely clouded over cornea, front of the eye, and quite small. I asked the parents if there was anybody else who had the same thing, and they said no. But Dr. Janssen did examine the eyes of both parents, and the father had, in the middle of the bottom, he had tiny cataracts. He was just 21. And when we asked about cataracts, he said, well, 3 of his sisters had had cataracts removed in their 20s or 30s and had really complicated surgery. And so we went to Placentia, about 2 hours away, and saw 13 of the 14 siblings of the father, all of their children, and really looked in detail at the size of the cornea, whether there was any clouding, whether there were any early cataracts, and indicated who we thought were affected. This is dominant, so somebody was affected could have affected children, but there was extreme variability, and this was very important to recognize. About 30 years later, a graduate student, Lance Toussaint, studied this family and identified the mutation, and we had been completely correct in determining who was affected, even though some had much more severe than others. Okay, next slide, please. Another condition we saw frequently early on was this condition called Bardet-Biedl syndrome, or BBS. And it was considered to be a rare condition, except for in Newfoundland. We had about 10 times the frequency. And there's a retinal dystrophy like RP, but there's also the extra fingers and toes, tendency to excess weight. The old literature all said mental retardation. There was early diabetes, there was kidney problems, and sometimes kidney failure at a young age. And we had over 30 families with BBS, and some of them had 2, 3, 5 affected individuals. And we didn't really feel that these people were mentally retarded when we saw them at the clinics and talked to them. They had very, very poor vision from a young age. And so we arranged to have clinical studies to find out the details of each of these features. I obtained an IQ test for those with visual impairment. And so it was all done by feel. So they had to feel if something, diagrams were different and so on. And the next slide, please. And from that testing, only 1/3 were actually mentally retarded. Others, in some cases, had learning disabilities. But others had completely normal intelligence. Some of them had been refused school because by grade 1, it was clear they had this condition, and the literature said mentally retarded. So this study with this large number of individuals, which showed the variability and the fact that it was not mental retardation. They had visual impairment. They were referred to the itinerant teachers, and some of them did extremely well and have very good jobs at this time. Next, please. And because of having so many families and large clusters, we thought we should be able to find the gene for BBS. Everybody thought there could only be one gene that could be changed to cause such a complex syndrome. But in fact, if you looked at all the families together, you couldn't figure it out. But later on, when the genetic methods became better, and one family was studied, and Michael Woods, a graduate student, was involved. And it was a Newfoundland family that found the first gene for BBS. And that gene, because of the type of abnormality it caused, led to the finding of really all the BBS genes. Next slide, please. So another condition that we saw frequently in the early clinics was something that we called Newfoundland rod cone dystrophy. There's night blindness from a very early age. It might be in childhood or teens, but central vision, so cones involved as well. But they had a very distinctive appearance of the back of the eye when Dr. Janssen examined, and that's how we, sort of grouped them together. And they all came from one area in the Conception Bay, so we had clinics there to see them as well. And from the pedigrees, the people were all related, although sometimes quite distantly. Next. And they had quite a significant visual field defect, so it was very close to the center of the eye, a very small central vision available. Because they were all in one area, we thought there would be just one gene mutated, and that everybody would have two copies of the same mutation. But when we first saw the family, it was too early to do molecular genetic testing. So the next slide. In the meantime, there were other families with RP that we saw, and there were individuals in 3 families that were seen at different clinics, but they all said that they lived in a particular small community on the northern peninsula. And interestingly, from the family history I obtained, there were some names in common. Next slide, please. And later, the church records for Newfoundland became digitized and became available for review. And a research assistant working with me reviewed the records and found exactly how these individuals were all related. And then it was possible to do genetic studies along with the group in Ottawa, Forge Consortium, and really across Canada, and found a very large deletion. And that was a mutation. So in this case, it wasn't like a spelling mistake. It was almost like a whole page missing. If you think of a gene like a book, a whole large section of the gene was missing. And every person who was affected had 2 copies of the deletion, the one from the mother and the one from the father. And we were able to study both parents in one case, they both had 1 copy. So they were carriers. And some of the brothers and sisters or children of affected people were carriers. So we expect that there might be others with RP in this area in the future, but they do all have a mutation identified. Next slide. And so these are each star, if you're able to see that on this map of Newfoundland is a different family with retinitis pigmentosa. There's families with the recessive, with dominant. There's 4 families with X-linked RP. In fact, there's 2 families that are with just about 20 or 30 miles apart. They both have X-linked RP, but they're different mutations. This young boy is actually part of one of those big X-linked RP families. His uncle was the first person we saw in our first clinic in 1978, and we followed the family for a long time, and he was at risk. And when the gene was finally identified about 30 years after we saw the first person, he was tested and he did not have the mutation, and some of his cousins at that time were being diagnosed with RP. But he wanted to do his science project on RP, so his mother drove him 2 or 3 hours up from the Burin Peninsula to St. John's so that I could go over the eye and how it functions and the problems with RP and so on. And so he could show this to his classmates, and it was the whole high school. He was in grade 7, and he actually won the science fair because he had such a good understanding that he could demonstrate to other people about the problems with an eye condition and the implications to the individuals. So next slide, please. Another condition that we have many families is called Stargardt disease or hereditary juvenile macular degeneration. So rather than age-related macular degeneration in the 70s or 80s and so on, this starts often, usually in the teens or 20s, sometimes earlier. And it's central vision loss. There's a particular appearance of the back of the eye. Next slide. And all of the families were in the eastern part of Newfoundland, and the majority of them actually come from one particular area of Conception Bay. And a research assistant at Memorial University did the initial studies with finding the mutations. There's only one gene for Stargardt disease, or for classic Stargardt disease, and it's called the ABCA4 gene, but it's a huge gene and has over 1,000 different mutations have been found, and it can be very difficult to study that gene sometimes. But we have found both mutations in most of the Newfoundland families, and there's 23 different mutations in the Newfoundland family, so it's extremely variable. The next slide, please. And so there's very specific implications of these genetic diseases in genetic isolates. There's different healthcare needs, and there needs to be dedicated diagnosis and screening. For instance, with the hereditary cancers, I set up clinical screening programs for early diagnosis of hereditary cancer, so there could be better outcomes. You want to have genetic diagnosis. In the early days, it was mostly done through research studies funded, for instance, by Foundation Fighting Blindness at the time, and so on. Now there is a medical genetics clinic within the hospital, and so that is responsible for doing most of the genetic diagnosis. It's also very important that there's education for the patients and their families about what the condition is, what the implications are, and what the inheritance is. But you also need to give it information to healthcare providers so they know what to look for, so that if somebody isn't doing well at school, think, could it be a vision problem? The public and other research need to know information as well. So the next slide. So I mentioned that I've gone into many communities for the rural clinics, and whenever I was there, I talked to family members and often went to the homes of older family members to talk to them. But then I received a knowledge translation award at one point, and what I did was then go to all of those rural hospitals to give talks, and I gave a talk to the healthcare professionals, and then I gave a separate talk to the family and public, and talked to individuals. Next slide, please. And so getting back to the Newfoundland rod cone dystrophy, it was distinctive looking and all in one area, and we thought there would just be one mutation, but 2 copies in each people. The next slide. But we did the studies, couldn't find anything. And then in 2000, researchers in Sweden reported a mutation in the gene, which is called RLBP1, in a group of patients. And Dr. Whalen, who was seeing the patients at that time, got archives of ophthalmology, came to his home every month, and he was reading it at breakfast, and turned the page, and it looked just like the back of the eye in our rod cone dystrophy patients. And so immediately our patients were tested for a mutation in that gene. And yes, they had RLBP1 mutation, but there were 2 mutations, which is why it was hard to find at first. Next slide, please. And so these are all the big pedigrees, and we seen some other families by that time some people had 2 copies of mutation A, some had 2 copies of mutation B, some had an A mutation and a B mutation. So as long as you had the 2 mutations, you had the same condition. Next slide, please. And later there were some other families found in other parts of Newfoundland, and we recognized them from the appearance of the back of the eye. Next slide, please. So this is just a summary of some of the conditions: Stargardt, Bardet-Biedl syndrome, rod cone dystrophy, RP, a chromatopsia, which is a problem with the cones and causes very early loss of vision and loss of color vision. Then the albinism, lack of pigment. Usher syndrome is RP plus deafness. And we have over 90 different mutations in different families. Sometimes in the same condition, there's multiple different mutations in different families. Sometimes, for instance, the RP, there's mutations in quite a number of different genes and also in a chromatopsia. So you keep having to work at every new family that's identified and try and classify, look at the clinical information and the molecular. The next slide, please. And so this is just different families in different places with different eye diseases. Next slide. And so it was mentioned that there's beginning to be treatment. So the problem is that there's a gene that doesn't work. And if you have something like RP, which is really the most common, the cells in the retina, that inner surface of the eye, normally the genes are working there to allow vision. And if the gene doesn't work, then the rods and or the cones deteriorate and you have a problem. But it's very difficult to get at those individual cells. And what people realized that they could do is, I guess the next slide, please. This is just the one diagram. It's like a great big spider web. And every one of those little colored marks is a different gene, and all those genes will be working together. And the thing is, this question about treatment is that the treatment is for a particular gene defect. And if you have 100 different genes, and for the eye, there's more than that, if you have to know which gene is altered before treatment is possible. And one thing that I thought was a very good lesson, not too long ago, I was at my exercise class. I came out, the car wouldn't go. I thought, oh good, I've got the cables. Somebody was nearby, he pulled his car up. We hooked up and it didn't work. Well, it turned out when the car was towed off to the garage that it was the starting motor. So it wasn't the battery. We could do something about if it was a battery problem, but it's just the same thing. If you have a gene, and we'll call it the ABC, and I don't mean the ABCA4 Stargardt gene, just one gene. If there is a treatment being developed for it, it's put in a new good copy of that gene. But you have a mutation in the XYZ gene. That ABC gene treatment is not going to help you. You already have a good ABC gene. You need a treatment for XYZ. And so if you look at all of these different genes, there are many studies that have to be done to develop treatments on the one hand, but then also we have to know what your gene defect is so you can match up. Next slide, please.
And I'm just going to jump in because I know we're running a little bit low on time. So
okay, there's hardly any more.
Okay, because I want to leave lots of time for questions.
Yeah. So when to develop gene therapy, a biotech company has to be aware and knows that there's certain different genes in the back of the eye that can be involved. And they decide that they'll work on this one. And what they do to get that gene into the retina is to put it in a virus. This is the time when the function of a virus goes into cells. It's causing a lot of problems right now in the world, but that feature of a virus has been used in a beneficial way. You put in the normal copy of the gene that can be altered into the virus. The virus, this is tested in an animal model to make sure there isn't harmful. You have to do studies to know exactly what the condition is like, what the vision is like in people. For instance, this Luxturna condition, that's for the RPE65 gene. You have to know what people's vision is like when they have that kind of mutation. Then people who are identified for treatment and they're given the treatment, and then you measure visual function and see if it's better, or at least if it stays constant and doesn't get worse, as was mentioned. So that the gene is injected under the retina, it goes into the cells, and hopefully it replaces the defective gene and works. Okay, next slide, please. So here it's being injected in, and next slide, please. And so you need a registry, whether it's in Newfoundland or elsewhere, for each of your patients with hereditary eye diseases. What's the clinical diagnosis and the details? What is the molecular diagnosis, which gene? Then you keep track of gene therapy clinical trials that are being developed or available in Toronto or London, England, or Chicago, and so on, and try and match up. Next. And so these are just a few. That Leber congenital amaurosis is an early type, very early type of RP. Some of the achromatopsia is the cone problem, X-linked RP, some of them, and so on. But there's many others that are being worked on, and it's a question of becoming available for patients. And so that's really, this is a nice, I think when you're doing research, it's always very important to have a plan. You're interested in the hereditary eye diseases, but you really don't know ahead of time exactly how you're going to gather the information. And you might be out there in one of those communities and somebody says something which turns out to be very key and makes you act a little bit differently in finding more information. So always be open to a new direction, and that, but keep on going. Okay, no, we don't need this.
Right,
so
great.
I hope that wasn't too long.
It's fascinating. It's so interesting. But I do know we have a few questions, so we'll jump right in. So I have a couple questions that I see have popped up in our Q&A. If you'd like to ask a question using audio, there's a couple of shortcuts you can use. So you just have to press Alt-Y if you're using a PC, or on a Mac, press Option Y, and that'll alert me to the fact you have a question, and then I can call on you when it's your turn to use your audio to ask a question. But let's get started with the Q&A. We have a question from Siobhan. She says, my name is Siobhan and I have RP, and my question is whether the Luxturna treatment will work with only RP65 or other types of RP, for example, 43.
It is a treatment specifically for the RPE65. And that's what my fancy spider web and the message about the battery in the car, that is specifically for one treatment. And there's definite reasons why you have to know exactly which gene, and that's the one you have to put in the substitute in the new good gene. So if you have a different gene that's affected, that treatment isn't going to help you because you already have a good copy of that gene.
Great. And another question from Milton. Milton asks, what is known about the heritability of AMD? My parents both had AMD, my identical twin and I have it, but my older brother does not. He says, thanks for the interesting talk.
Well, even if there was inheritance of some types of RP where we know the gene defect, some children might have it and others not, just by the way you inherit. AMD, many people have looked at it, and it certainly clusters in some families, but there's no direct evidence that there's single genes that can be inherited, like the RPE65 gene or the rhodopsin gene and so on. It seems that it's more several different factors together, and even things like blood pressure and so on are affected by these multiple different genes clustered. And so it can look in a family like it's inherited, but there's no gene to test and say, oh, older brother doesn't have this particular change, and you know, and the other two brothers do. There's nothing that anybody knows to look for as a single gene. It seems to be a combination of things. People are working, and hopefully there'll be more knowledge in the future.
Great. Marie asks, is anyone testing therapy for Usher's?
Usher's syndrome, again, is not just one disorder. There's actually Usher 1, Usher 2, Usher 3, and there's probably 12 different genes that are known. And one or two of them, I think people are working on. But again, it's critical which type of Usher, which gene is defective in the individual's case with Usher syndrome. So it's just like with RP, a certain one might have a treatment and another one not.
Okay, Mary Kate asks, she says, I have RP and had genetic testing done last year. They were not able to find the RP gene, but they did confirm that I'm a carrier for OCA2, autosomal recessive. Is there any benefit to registering this finding anywhere for research purposes, even if I don't have the condition?
So presumably that she has one copy of an OCA2 mutation.
I'm not sure.
Did she say she's a carrier?
She says the carrier for OCA2.
Yes. So that would be, that's the term, genes are in pairs. And for OCA, you need 2 altered copies to have the disorder. If you have just one copy, then you're called a carrier, but the other gene is doing the job. And so you can't look at somebody and tell if they're a carrier or not, and everybody is going to be a carrier of something. So I think it's something to keep in mind, but it's not something she could be treated for or needs to be treated for. It's very different from the RP. And unfortunately, as I can remember when there was 20 genes for RP and they thought, well, there'll be a few more. And now there's hundreds of genes and there's still more to be found. But she should have genetic testing again in another 2 or 3 years, because by that time, her gene may be identified.
Okay, great. And this is a question from Rebecca. So Rebecca asks, if a single genetic mutation, such as to the BUX-C1 gene in Axenfeld-Rieger syndrome can cause issues in multiple areas of the body, such as glaucoma in the eyes and enhanced risk of cancer. What is the best gene therapy approach? Should the eyes and body be treated in a single therapy or separately? And is gene therapy and editing effective treatment, or can it cause other issues?
That is complicated. That any of these conditions where there's multiple parts of the body potentially involved can be very difficult. If it's just in the eye, you can do that type of injection like I mentioned, and it goes into the cells of the retina. But if another part of the body that's involved is, say, muscle, and there are conditions that might be eye and muscle, putting the normal gene into the eye isn't going to do anything to do with the muscle part, or in Bardet-Biedl syndrome, the extra fingers and toes. So it's much more complicated when it's a multi-organ problem. Sometimes, if it's very early, there might be some kind of a medical treatment, but I don't know of anything, for the whole range of problems with Axenfeld-Rieger.
Okay. And notice we have someone who's put their hand up, so let's try to answer that. So it's Mike Hokovar. And so Mike, I think you just have to unmute yourself now. So you just press Alt-A on Windows or Shift-Command-A on a Mac. So let's see if we can hear from Mike. Are you there, Mike? Okay, well, maybe we'll do another question and come back and see if Mike can figure that one out. So let's see here. So Linda asks about your spider web sort of diagram there. She said, how do they determine which gene in the spider web is the problem gene?
Okay, the spider web is put together afterwards once you know about the gene. When you're finding it's done through a genetic testing, usually a genetic testing company, and they'll have either they have a panel of all the genes that are known, and they test all of those and say which one is it. But then they were finding, like some people said, that doesn't find anything. And so what is done nowadays is they sequence from start to finish all the DNA. And so this is like taking a very large book in the library, and you've got to read every word of every page and see if you have any change. And that's really what you're doing, but you're doing it by a method of sequencing. And the trouble is, it might as well be in Russian. So it's not just that you have to find a change in an English word. You're trying to find some very, very subtle change. And so they're finding more and more alterations, but sometimes it's hard to recognize what is a mistake. Some people will write a sentence and put a comma somewhere, and somebody might not use a comma there. So is that really a mistake or not? It's not always easy, but the people on the molecular side of things are developing better and better analysis, but the spider web is really put together afterwards. Once you've found all the genes, then other people see how they're related, but you do it by looking at the DNA.
Great. Do we have Mike now? Mike, can you hear me? Can you speak? I see his name's popped up at the top here, so I didn't know if we could. Nope, I still know Mike. Okay, so let's try another one. So Dora asks, are you aware of any genetic research done on the EYS gene?
Yes. Now, I was at a meeting a couple of years ago when somebody was working on the EYS gene, and I am not, it's something I could look up and possibly send an email to Morgan, and that could be
Sure.
passed on. Exactly how much is being done, I don't know, but I do remember that, somebody was talking about that gene, and that was their interest. It's a fairly commonly mutated gene in RP.
Okay, perfect. Well, we'll reach out to Dora with a bit more information after. I have another question here. Are RP and glaucoma related by the same gene defect?
No. RP isn't a change of the cells in the retina. Glaucoma is a problem of pressure in the eye, and it's really in that round circle of the eye. Glaucoma is when there's too much pressure, so it's a different mechanism. It's not by individual cells, not in the retina. There may be certain circumstances where certain types of retinal problems might relate to glaucoma, but in general, glaucoma is a different type of problem. There are genetic causes of glaucoma, but those are different genes.
Okay, great. We have some questions that came in by email. Elaine asks, do you have any idea of a timeline leading to a genetic cure for retinitis pigmentosa? My son, who's age 45, has this disease, and potentially one of his daughters who's 11.
Okay, this is difficult because the thing is, which gene, this sounds like it may be a dominant if he's affected, and possibly his daughter. Is that really what you'd said?
Yeah, I think just looking sort of for like a timeline, if there's going to be some sort of therapy.
Nobody really knows the timeline. It's because there are so many different genes, and whether the gene that is known in that case, if it's not even known yet, then there's going to be a delay before the gene is known. Then some company has to decide to study that gene and develop a treatment and go through clinical trials, go through the animal models and then the clinical trials. At the best of things, it's several years, and that would be on the assumption that the gene is already known. But it's not quick because you have to go through so many steps to make sure of the safety.
Absolutely. Okay, so another question from email. Leanne says, she said, Dr. Green, you met my young son in the mid-90s. He was the first patient to be diagnosed with BBS at BC Children's Hospital. Accessing worldwide research studies has made it possible to approach his multiple health challenges in an efficient manner and provide him with a quality of life. Now that BBS has been further divided into BBS 1 and BBS 2, which organization?
About 20 actually.
20, sorry, BBS 1 to BBS 21. Which organization do we contact to establish which BBS number he falls under to help us with future life planning?
If he's had molecular testing, whether it's BBS 1 or 2 or 6, is dependent on which gene is found to be altered. So the molecular testing determines that. And I don't know the details of management, but the first step is to make sure that the molecular testing is done and the mutations in which gene are found.
So even like you said before, like if he's had the testing prior, he should probably get it updated, right?
Well, if he's had the testing prior, it may be known. And the thing is, they're called 1, 2, 3, which is easier, but each of those genes also has a gene name. And some people may use, for instance, rhodopsin in the RP. Rhodopsin is the name of one of the genes, but that's also called RP and such and a certain number, but there's so many with RP that you don't use the number so much anymore. But so it may be known, and it's just that somebody has to look at a genetic test report and see if there's a definite answer. If not, it should be tested again. If it is, then a genetic counselor could explain exactly what the testing means. Is it known which of those BBS genes it is?
Yeah, and if you have questions about getting genetic testing in your specific province, you can always reach out to our health information line that Larissa mentioned at the beginning. We have some resources that can help, because every province is a little bit different about how to get genetic testing. So we can sort of help guide you through that process a little bit. I know we're over time. There's so many more good questions. We'll do a couple more, and then we'll, if we don't get to your question, we'll be sure to reach out afterwards with some answers. So, okay, let's take some more from the chat. So Lindsay says, I have a GUC1A1. Where would I go to try and find research or studies? We only found one study on this, and it was in China, and I was unable to get a hold of their researchers. I would like to learn more information about my eye disorder, but unsure where to go to look. Thank you for the presentation.
Could you say that again?
Sure, it's GUC1A1.
Yeah, GUC, yes. Okay. Presumably that's an RP, but did she say? I think I've heard of that, and I think there was, maybe she put me in touch with her, and I'll try and help a little bit.
I'm muted. Yeah, we can reach out to you, Lindsay, and try to find some information for you. Okay, let's see what else. We have so many more. Okay, so Brenda asks, I did the genetic testing last year, and they identified I have PRPF3, formerly known as HPRP3. How do I register for clinical research?
This is where you need your assistant, which I don't have these days. The important thing, and in that last slide I said, you need to find out, everybody in Newfoundland or wherever, what mutations they have, if you can possibly find out. And then parallel to that is to find out all of the testing and treatments that are going on around the world. And some labs in Oxford, England has a lot of things going on. There's certain groups in Germany who are working on other conditions. It's almost like a full-time job to keep this up to date. And this is where, if only there had been an ARVO, which would have been like last week, you would have gone to all the posters and talked to everybody, or I would have, and other people, and keep putting together a list. I think this is what we have to do as a community, is keep putting this together, because there's new things happening all the time in all different places around the world. But what we need is that for the clinicians and the geneticists and the families to be able to go to something that keeps updating a list.
Absolutely. I know we have a lot more questions and we haven't had a chance.
Morgan,
I was going to say, Larissa, do you want to jump in?
Yeah, can I just jump in really quickly about that? Dr. Green's right. It's such a complicated, potentially full-time job. There is a website called clinicaltrials.gov, which is, I wouldn't say the easiest website to use, but you can look to see what trials are available with a specific disease or a mutation. And again, we keep saying this, if you've got questions, our health information officer can at least help walk you through the website. Another way that we are suggesting is for patients to join the Fighting Blindness Canada patient registry. And so our goal is to try to keep patients up to date about new trials, especially if they're coming to Canada. If a child comes for either your indication or your disease or your specific genetic mutation. So those were, I would say, the two things I would add to Dr. Green's response.
Right. And so tomorrow there is a webinar of people involved in the registry, and will be, I mean, I will bring up some of the points that were discussed today, but everybody from different angles is trying to get this information together. The thing is that the research is moving quickly, too slowly, you know, in some ways, but there's always new things coming up, and there's so many different labs with different types of expertise just working on different things that it's hard to keep a totally up-to-date resource, and a resource that both clinicians and scientists and the public can understand.
For sure.
But that's being worked on.
Yeah. So I think we're going to have to leave it there, unfortunately. I know there's a lot of questions that we didn't get to, but I have a copy of all of them, and I'll work with Dr. Green and our health information officer, and we'll make sure to get back to everyone. One of the questions was even, would it be possible to just have a Q&A webinar with Dr. Green? I think we could all spend a lot of time talking together with Dr. Green about this.